Hi, everyone. My name is Molly Murphy, and this is my presentation in regards to the epidemiology, pathogenesis, history, and associated details to the disease commonly known as anthrax. So I'm going to go ahead and share my screen. So this is my presentation I put together to hopefully teach you a little bit more about the condition known as anthrax. So I like to start off with some objectives which describe what the audience should be able to accomplish at the conclusion of this presentation. So by the end of this presentation, you should be able to describe the condition of anthrax itself, including associated details such as the history, the discovery behind the infection, the taxonomy, how is it categorized, what other infections and microbes is it similar to, its morphology, its characteristics, structures, and its shape, pathogenesis, which is how exactly it's spread, and the epidemiology. At the conclusion of this presentation, the audience should be able to connect the information obtained during the presentation to describing necessary treatment and prevention plans. So kind of connecting what you learned from this discussion to how you can protect yourself, how you could protect others, and how you could practice appropriate prevention plans. So kind of connecting this to real life is important. And the audience will also be able to comprehend visual guides. So throughout the presentation, we're going to analyze graphs, other illustrations, and you should be able to connect the knowledge you obtain to interpreting whatever the image is showing you. So beginning with the discovery of the condition and the history associated by it, and ultimately just discovering anthrax. It was first a disease of man and animals shown to be caused by a microorganism. So it was one of the first diseases that was connected to being caused by a microorganism. Hindu, Greek, Roman literature and philosophers wrote about anthrax, and more specifically how it targeted domesticated but also wild animals. So a lot of the cattle and other animals were becoming sick. So it's interesting to kind of build the connection between what we know now and way back what occurred throughout worldwide history, to kind of uncovering that the condition anthrax was what was affecting these cattle that they wrote about in their philosophical books. So it was the disease of which much of the original work of bacteriology was done, and from which many well-known principles of microbiology were derived. And it's spread throughout every country, pretty much worldwide, including early Egypt, Italy, Greece, where it killed many livestock, but also humans. So this kind of began the thoughts and the discussion behind, can this microorganism infect livestock, infect animals, and is that how it's being transmitted to humans, or are humans contracting the infection? So early on, people kind of realized the connection between that this microorganism can infect man, but also animals. At the time of Pasteur's experiments, Robert Koch had concluded similar work. He worked out the complete life cycle of the bacillus. So a bacillus is a rod-shaped bacterium. A bacillus is a collective term used to describe the shape of a bacterium. So using Koch's postulates, Pasteur's experiments, they began to pretty much map out the complete life cycle of this bacteria and how it begins infecting livestock and humans. The now famous Koch's postulates were actually proved by anthrax, so the transmission of anthrax, and Koch's very famous postulates were proved true through the transmission patterns of the bacterium that results in the condition known as anthrax. He's also been claimed to have been the first to prove how anthrax happens, and he's sometimes supposed to have done this in his 1876 paper by using the causal criteria that later began to be now famously known as his postulates. So here is kind of a overview timeline of the discovery of anthrax. So 700 BC, ancient origins of anthrax. People started writing about livestock being infected, humans being infected. And it wasn't until 1752 where the first clinical descriptions of the disease came about. In 1877, Robert Koch develops anthrax and uses the transmission patterns and symptoms and the associated characteristics of anthrax to prove his postulates as being true, which now, present day, his postulates are very famous. So it's kind of interesting to work that into the discovery of this bacterium that causes anthrax. In 1881, Louis Pasteur created the first vaccine against the bacterium. 1937, the anthrax vaccine for animals was the vaccine was used in veterinary offices or whatnot, but it was used in mainly animals. And the transmission pattern, people noticed that when the vaccine was delivered to animals, humans were less likely to contract the infection. So that kind of allowed for more discovery of details associated to the transmission pattern of this infection. 1960s, the global concern about the use of bioweapons. So I'll talk about this a little bit later on, but anthrax is a bacterium. It produces spores, endospores, and these spores can live in very harsh conditions for a very long time, up to 10 decades. So once people started to realize this, they use this for a negative cause. So they kind of use this as a bioweapon. Bio, I like to tell my students the prefix bio means living. So this was used as like a living weapon in biological warfare because scientists and other individuals began to notice and realize that these spores can live out in the air and in the soil for a long period of time. So they used that for a negative reason for biological warfare. 2010, doctors find a new form of the condition, which is very common. We call these new forms of different infections, we call these strains. So very common for the flu to develop different strains. These are basically just little mutations, how pathogenic either microorganisms or viruses mutate, and natural selection favors these mutations. And it's pretty much how they're able to change a little bit to overcome your immune system. That's a whole other topic. In 2001, anthrax attack on America. So this is kind of the history, not up obviously to present day, but kind of an overview of the discovery and transmission patterns of the bacterium and anthrax. So its scientific name, it is a bacillus, which means it's a rod shape, like I mentioned before. And its scientific name is Bacillus anthractis. And when you look under the microscope, you can see these rod shapes that are kind of highlighted in this image. So I thought this was a nice visual of the condition of the bacteria, but also connecting the shape of the bacteria to why it's named scientifically the way it is. So here is also, you could see in this number 1 box, the scientific name of the bacterium that causes anthrax. Anthrax is the common name of the condition. The Bacillus anthractis is the bacterium that causes the condition. So it is a bacteria. Most bacteria are prokaryotic, which means they don't have that membrane-bound nuclei. And specific characteristics of this bacteria is that it produces dormant spores, which means they're not active, but they contain little pieces of genetic material that can live in the soil for a very long time, up to even decades of time. And when herbivores like this cow you see on the screen eat the spores that are living in the soil, that's how they become infected. So when spores get into the body of an animal or even a person that may have come to contact with the spores in the soil, a place that's rich with water, sugars, and other nutrients, these spores can become activated. So the bacteria can become activated and start dividing. And now we have an active growing, active culture of cells that are pathogenic. When they become active, they multiply, they spread out in the body, and they produce toxins that cause severe illness and, in some individuals, unfortunately, death. So we'll talk more about the illness component here and how the immune system pretty much targets the toxins that are produced by the Bacillus anthractis. So the structure, I mentioned this a little bit before, but it's a member of the Bacillus group, which means it's rod-shaped. It's a collection of a gram-positive spore forming, obviously, to spore forming. We just talked about that's how that relates to its pathogenesis, how it can infect multiple individuals and even livestock. And they are facilitative, facultative, and aerobes, which means they don't require oxygen to to perform cellular respiration, which makes sense because these spores can live in the soil for up to decades, up to 10 years. And depending on what soil where these spores are located, there may not be an abundance of oxygen supply. So if there's an area that's rich in waters, sugars, and other nutrients that's listed here, it can still perform cellular respiration without the presence of oxygen and be living. The bacteria, Bacillus anthracticus, produces toxin proteins during infection of mammalian hosts. That also was listed on the image in the last slide. When the bacteria enters either the livestock or a human, that's why it's summarized as mammalian hosts. But when the bacteria infects a host, it secretes toxins to the immune system to begin its infection pattern. So here's another image of the bacterium itself. So this is a singular bacterium. And here you could see typically what the culture or multiple bacterium would look like under the microscope. So kind of just to go back for a second, if you look at this picture, this is a sample of the Bacillus anthracticus. You could see its rod-like, string-like nature. So that's what this illustration here is representing under the microscope. This is typically what you would see, and you could see that these bacterium are rod-shaped. To look more at the internal structures, you can see they have an endospore, they have spore DNA, they have their DNA, it's prokaryotic, like I mentioned before. So the bacterium itself, its DNA is distributed throughout the cytoplasm. It's not compartmentalized by a nucleus. All right, so this is not the nucleus, this is the endospore and separate spore DNA. So once these spores are distributed in the soil or in the air, they can cause infection. You can see it has the structure, the bacillus has a cell wall, it has a cell membrane, cytoplasm, which is just the free material, ribosomes. So this is just the general structure of the singular bacterium itself. And then this is typically what the infection would look like under the microscope. So the pathogenesis, how exactly does the Bacillus anthractis cause anthrax? So the disease is initiated by the introduction of the endospores into the human body or into the body of livestock. Experimental results from early studies on anthrax suggested that in addition to the capsule, Bacillus anthractis expressed a toxin that was a major virulence factor. So when the endospores enter the body, they release little toxins to pretty much combat the immune system of the host. And that is a factor that's used to determine whether or not the person is infected with anthrax. But typically the infection begins with the endospores entering the body. And again, those spores can live in the soil, can live in the air in the soil mostly for up to 10 years, which is 1 decade, which is a long time. So typically herbivores will be grazing the farmland or the grass in general. They consume the endospores, they infect them. If people eat an infected, let's just say cow, they can obtain the infection, or people can directly come into contact with spores themselves and become infected. So spores can live in the soil for many decades. Herbivores consume these spores, they become infected. Human exposure to the infected animals can result in themselves being infected. Vultures can actually physically spread the organism throughout the environment. So vultures are animals that consume dead animals or dead material. They break down organic material similar to how decomposers would. We call these scavengers. But if they come into contact with the spores, they can actually physically move the infection throughout an area, similar to how a vector would spread, like how malaria is the condition that's spread by a little protist that lives in the gut of mosquitoes. So mosquitoes are the vector that's spreading the infection. The mosquitoes themselves don't feel sick, but they can spread the protist. And when they bite their host, the protist can move from their gut into the host, and that's a vector spreading the infection. So similar idea here, how vultures can spread the spores of Bacillus anthractis and cause anthrax. And it's also the single greatest biological warfare threat. So that's another way that it's spread. So here, these 3 bullets describe how exactly the infection is spread through a population. So in the first bullet, it says that anthrax spores can live in the soil for many decades, but like the image overviewed, this is specific to certain conditions, obviously, that keep the spores alive. So such conditions include rich in organic matter, a pH of about a 6. It can also withstand dramatic changes in climate, such as abundant rainfall or the opposite, prolonged drought. These spores are designed to live in very drastic conditions. Spores that can be sprayed in the air and infect individuals. A World Health Organization report estimated that 3 days after the release of about 50 kilograms of anthrax spores along a 2-kilometer line upwind of a city of about 500,000 population, 125,000 infections occurred, and that resulted in 95,000 deaths. So yes, these spores can live in air droplets in the soil, and that's mainly why they're used as a biological warfare threat or device, which is scary. Here's another image that kind of overviews and allows you to kind of visualize everything that we talked about in the last slide and kind of overall, in specifically to regards of the epidemiology of anthrax and how it spreads. So it's an ancient zoonotic disease, which means it can spread and it can infect animals and it can infect humans, which primarily infects herbivores, with humans occasionally being infected. And we talked about why herbivores or plant-eating animals are mostly infected is because the spores can live in the soil and the herbivores eat the grass and come into contact with potentially infected soil. It remains a problem in low and middle income countries, not too prevalent in the United States. So this, I'm not going to read through this entire thing because we kind of overviewed it in the last couple slides, but this kind of shows you how exactly the condition spreads, which a fancy way of saying that is the epidemiology. So you can see the germination of spores to develop in vegetative forms, multiplication in lymphatic spleen results in death. Ingestion, so if the herbivore, like the cow, ingests the spores through grazing, browsing, drinking, then the germination of the spores could lead to death. The spores mainly in humans infect the respiratory system, but also the gastrointestinal system, and can lead to other scary things like meningitis or menincephalitis, which is basically, excuse me, fluid buildup in the meninges, which could be deathly. Or it can be cutaneous, which means, and this is most commonly how it presents in humans, without getting into too much detail of symptoms, because this is mainly about how the infection spreads. But if you do contract the infection, it mainly presents itself cutaneously, which means on the skin, and you'll see some sort of lesion. And that's pretty much a symptom associated with being infected with anthrax. So treatment of those few symptoms I mentioned, there's therapies with different antibiotics, but also respiratory therapies that help with anthrax that's been inhaled. If you've inhaled it, if you've come into contact with it through touching it or whatever, ingesting it, whatever that is. There are penicillins, amoxicillins, other antibiotics that have been synthetically created to combat the infection. Controlling anthrax in humans depends primarily on effective control of disease in animals. So animals are mainly the ones that contract the infection. So if we can control them becoming infected, then it's less likely that they'll transmit that to humans. So like I mentioned early on in the discovery of the bacterium, the microorganism, people were vaccinating animals. So this has been an effective treatment option. So the treatment will kind of vary depending on how you've become infected. How can we control and prevent the infection? We can control the disease through proper use of vaccinations, countermeasures designed to risk the exposure to a contaminated source. So being aware of how you can contract the infection, proper use of treatment. So if you are infected, obviously get yourself antibiotics so that you don't spread the infection further, eliminate contact with infected animals, like we talked about before. Vaccinations for animals are effective. All these will help prevent the infection, but fortunately enough, it is not so common in the United States. This graph here shows the effectiveness of the vaccination. So you could see here's the timeline. So from 1984, how many livestock were infected by anthrax. You could see in 1984, there were like 7 up to 8 million. I think this is a million, yeah, livestock infected. And then you could see as time progresses, and the doses of the vaccine have been distributed, there are less infections. So in 2011, there's 0 infections as the vaccine has been distributed. So that's interesting. And then the last 2 slides just have all of my references, resources, citations of images. So just to go back to our objectives, hopefully at the end of this presentation, you can now describe the taxonomy, taxonomy of the anthrax, the fact that it is a bacillus bacteria, its pathogenesis, how it spreads, and its epidemiology, how it spreads through larger populations, and more so what happens if you become infected with it. You should be able to describe certain treatment plans that have came about and how we can prevent the infection. And hopefully you're able to describe the visual guides that were included in the presentation. So I hope you learned something about anthrax, and I hope you found this interesting, and I really appreciate you listening to my presentation. Thank you.